Okay, today we are discussing the question from 59th question. The development of fruit without fertilization is called as. So we know different kinds of fruit formation. One is, see here, there are some options given: triple fusion, double fertilization, syngamy, parthenocarpic. So these are the options given. నాలుగు ఆప్షన్స్ ఇచ్చారు. త్రిసంయోగము, ద్విఫలదీకరణము, సింగమి, పార్థనోకార్పిక్. So the development of fruit, usually the fruit develops after fertilization. That is natural. So once the fruit is fertilized, what does it mean? That means the ovules, in the ovary are fertilized. The ovules in the ovary are fertilized. So the ovary becomes the fruit. We already discussed about this, how the ovary develops. So post fertilization and pre fertilization changes. There are some changes post fertilization and pre fertilization. After fertilization, what happens? You see, before fertilization, see, this is the ovule. For example, this is the ovule present in the ovary. The entire ovary becomes the fruit. The ovule becomes the seed. Ovule becomes the seed. And ovule integuments, they become the seed coat. Nucellus, which is diploid, it becomes a perisperm after fertilization. And female gametophyte, it is also called embryo cell. And egg cell in the egg apparatus, after fertilization, it becomes a zygote, that is diploid, and it is developed into diploid embryo. And central cell, we already discussed. Central cell, it contains two polar nuclei. Okay, and after the fusion with the second male gamete, that is called as triple fusion or double fertilization, it forms the triploid primary endosperm nucleus, that is also called as endosperm later. So these are all the before fertilization and after fertilization changes. So now the question is development of fruit without fertilization. How the fruit is developed? It is developed from the ovary. The entire ovary with the ovules, it is transformed into the fruit. The ovules within the ovary becomes seeds. The ovary becomes the fruit. So here the question is, abnormally, there is no syngamy, there is no double fertilization, there is no triple fusion, but it becomes a fruit. What is that phenomenon? Is called as parthenocarpy. Okay, the development of the fruit without fertilization is called as parthenocarpy. It is one of the most important questions. Okay, and one more question. Usually there will be some confusion between the parthenocarpy and parthenogenesis. There is a difference between the parthenocarpy and parthenogenesis, because one of the doctors, he told me once, he wrote an examination for the government doctor post. So what he told, once I visited and he was asking me, I got this question, what is the difference between the parthenocarpy and parthenogenesis? He asked me the answer because he was confused. So parthenocarpy is related to the plants, related to the development of the fruit without a fertilization. But parthenogenesis, it is mostly related to zoological term. It is the development of that organism, animal organism, from the egg cell, because in the animals they lay the eggs. After fertilization, that egg is developed into an organism. Okay, So in case of the animals, we usually refer parthenogenesis, that you should remember. And in case of the plants, for the development of the fruit, we use the term parthenocarpy. So that you have to understand. So next question, the eukaryotic ribosomes consist of the subunits. You know, there are two types of the ribosomes. What are those ribosomes? 70S ribosomes and 80S ribosomes. Usually we find 70S ribosomes in the prokaryotic organelles. Prokaryotic organs అంటే కేంద్రపూర్వక జీవులలో ఉండే రైబోజోమ్లు 70S ribosomes. In case of the eukaryotic organelles, we find the ribosomes. These are called as 80S ribosomes. ఇక్కడ ఏమడిగారంటే what are the subunits? What are the subunits of the eukaryotic ribosomes? First we should know what are the ribosomes, what kind of ribosomes present in the eukaryotes. 80S ribosomes. So what are the subunits of 80S ribosomes? That is 60S and 40S. 40S ribosomes, these are called as subunits. Small subunit is called as 40S. The large subunit is called as 60S ribosomes. So there is a sedimentation coefficient. What is this? It reflects the sedimentation coefficient. So when 40S and 60S subunits are united, it becomes 80S. It is not 100. It becomes a 80S ribosome. So those subunits also called as Swedberg units. Okay, it reflects the sedimentation coefficient of the ribosome. So one more thing, another question, important question is, there is one mineral element required for the union of 40S and these two subunits. Two ribosome subunits need one mineral element. What is that mineral element required for the union of these two subunits? No, that is magnesium, Mg+2. Mg+2 ions are required for the union, for the fusion of these two subunits. Then only they form a 80S ribosome. And in case of the prokaryotic organelles and also in case of the organelles, cellular organelles, like mitochondria and chloroplast, also we find 70S ribosomes. Okay, semi-autonomous organelles, these mitochondria and chloroplast, they are called as semi-autonomous organelles because they have some specific features like the presence of the circular DNA and also presence of the 70S ribosomes in the mitochondria and chloroplast, and also prokaryotic organs. So they have 70S ribosomes. And what are the subunits of 70S ribosomes? The small subunit is called 30S ribosome. The large subunit is called as 50S ribosome. So when 30S plus 50S ribosome, they are united, they form 70S ribosome in presence of magnesium, and that means Mg+2 ion. Okay, so this is very important question. And next, the pollen is shed and carried to the stigma in a, what is the stage of the pollen when it fertilizes the ovule? Okay, so usually after the pollination, what is the pollination? When the pollen reaches the stigma of the ovary, that process is called as pollination, whether it is the self pollination or cross pollination. When the pollen attaches, when it reaches the stigma, that is the tip of the style. Okay, ovary contains a style and stigma. When the pollen is fallen on the stigma and when it is compatible to that species, it germinates and it forms a pollen tube. Through the pollen tube, what happens? The pollen, it divides, the nucleus in the pollen divides, and finally it forms two male nuclei. Okay. So at what stage the pollen reaches the stigma? That is a two cell stage. Okay. Next. So one is a vegetative cell and another is a generative cell. Next, the question is the cell wall of plants is comprised of. Now we came to cell biology, cell wall structure. And in the cell wall, what we find usually middle lamellum and primary cell wall, secondary cell wall. Since they are asking in general, what are the components of the cell wall? Glucose and racin, it is wrong. Fructose and tannin, it is wrong. Starch and suberin, it is wrong. And hemicellulose and pectin, it is the right answer among these. What is the best? Sometimes they may give cellulose, hemicellulose, and pectin. So usually, what are the components of the cell wall? In the primary cell wall, we find cellulose, hemicellulose, and pectin. And in the middle lamellum, calcium and magnesium pectate. If it is a secondary cell wall, we see there is some like chitin or lignin. These are the cell wall components in the secondary cell wall. So since it is not specific question, they just asked the components of the cell wall. They did not specifically ask whether it is the middle lamellum or primary cell wall or secondary cell. If they ask about the secondary cell wall, we have to say lignin, chitin, etc. But they asked in general. So out of these four choices, we have to see the one of the what is the best answer, that is the hemicellulose and pectin. Next question is Singer and Nicholson proposed the membrane theory of. So there are several theories proposed. Okay, different theories are proposed regarding the cell wall. This one. So just I have given some explanation for you. Not only this, see. Yeah, okay. We just I will explain you. I don't know. So here you see Singer and Nicholson, what they proposed. So to explain the cell membrane, there are so many theories proposed. One of them is sandwich model and fluid mosaic model, unit lamellar model. Okay, so fluid mosaic model was the most popular and accepted by everyone. This model, it was proposed by Singer and Nichols. Okay, and unit lamellar model, this is a unit membrane model also it is called as, it was proposed by Robertson. This is also very important answer. Okay, unit lamellar model, who proposed this unit lamellar. So next, the powerhouses of a cell are, there is a chloroplast and mitochondria, Golgi complex, and ribosomes. Ribosomes are useful for the protein synthesis. Chloroplasts are useful for the synthesis of the carbohydrates or glucose. And Golgi complex is required for the secretion function. For the secretion, it is important. That means intracellular secretion. It is also called the main function of the Golgi complex is secretion. That means Sravakriya and good. Chloroplasts, they are useful for the photosynthesis. They prepare the food in the form of the glucose, fructose, and later it becomes starch. And ribosomes, they are required for the synthesis of the proteins. Polypeptide synthesis is the called. So now we come to mitochondria. In the mitochondria, what happens? The energy is released from the mitochondria in the form of the ATP. How? Because there are two process Krebs cycle. takes place. After the, we see in the respiration, there are three main processes in the aerobic respiration. Okay. In the aerobic respiration only, we find the mitochondria. Okay. In anaerobic respiration, there is only one process takes place, that is glycolysis. Glycolysis occurs in the cytosol or cytoplasm, glycolysis. Okay. And after the glycolysis, what happens? The end products, that is pyruvic acids. Okay. They are converted into acetyl CoA and they reach the mitochondria. They participate in Krebs cycle in the mitochondria, in the matrix of the mitochondria, where Krebs cycle occurs in the matrix of the mitochondria. And later, the products which are produced during the Krebs cycle, they participate in the next cycle, that is the electron transport. Okay, that is also called as oxidative phosphorylation. It takes place in the inner membrane of the mitochondria. They are also called as cristae. In the inner membrane of mitochondria, what happens? ATP is produced. That is called as energy currency. ATP is called as energy currency. So mitochondria is responsible for the synthesis of the ATP, that is energy currency. So it is required for the functioning of the organism. So it is called as powerhouses of a cell. Just like the electricity, we need electricity. Just each organism require energy in the form of the ATP that is produced during the oxidative phosphorylation in the mitochondria. Okay, right. Next, electron microscope was developed by. So electron microscope, it is very important. The answers given are Knoll and Ruska, Jacob Monod, Schwan, Brown, and Hooke. So here we see R. Brown and Hooke. R. Brown, Robert Brown. So Robert Brown, he discovered nucleus, and Robert Hooke, he discovered cell. Okay, initially, and M.J. Schleiden and Schwann, what they discovered? Cell. They proposed cell theory. Schleiden and Schwann, they proposed cell theory. That means cell is the structural and functional unit of the organism. That is the cell theory proposed by. And next, Jacob and Monod, what are their contributions? Okay, so they proposed operon model. What is the operon model? In the molecular biology, in the genetics, you read it, operon model. Okay, that means regulation of the gene regulation, in the prokaryotic organisms. Okay, all the whatever the genes which are all are attached. Okay, so that is called as operon model. There is no splicing in the RNA, mRNA in the prokaryotes. So the operon model was proposed by Jacob and Monod. And also one more thing, mRNA was also explained and discovered by Jacob. Next, we come to the right answer, that is Knoll and Ruska. They developed the electron microscope. Knoll and Ruska. So Ruska, sometimes we can use any word, no problem. So you remember this electron microscope. Next, the primary function of rough endoplasmic reticulum is. So endoplasmic reticulum, yes, here you see, this is endoplasmic reticulum. This is the nucleus. There is a nuclear membrane. In the eukaryotic, only we find this structure, because the nuclear material is always surrounded by the nuclear membrane in the eukaryotes, and there we find the nucleus very clearly. But in case of the prokaryotes, there is no nuclear membrane surrounding the nuclear material. There is only nuclear material, but there is no specific identity like a nucleus in case of the prokaryote. So here in the eukaryotic organisms, there is an extension of the nuclear membrane. From the nuclear membrane, there is one network-like structure is produced, that is called as rough endoplasmic reticulum. From the rough endoplasmic reticulum, one more network-like structure is developed, that is called as smooth endoplasmic reticulum. What is the difference between the rough ER and smooth ER? There is only one difference, that is the ribosomes present, they are attached to the surface of the rough endoplasmic reticulum, rough ER, but there are no ribosomes attached to the smooth endoplasmic. So the main function of the rough endoplasmic reticulum, due to the attachment of the ribosomes, what is the main function of the ribosomes? We already discussed, protein synthesis. Okay, due to the presence of the ribosomes, the main function of the endoplasmic reticulum is protein synthesis. Okay, so it is not chlorophyll synthesis, it is not carbohydrate synthesis, because carbohydrate synthesis occurs in the chloroplast only, because glucose is fructose, and it is converted into glucose. Glucose is converted into, it is after polymerization, it forms the starch, that is a carbohydrate. It occurs in chloroplast. Okay, lipid synthesis, it is also wrong answer. So the right answer is protein synthesis. Rough endoplasmic, the primary function of rough endoplasmic reticulum is protein synthesis. Okay, next question is, a nucleoside consists of. So what is the nucleoside? You just see the DNA structure. In the DNA, what are the components of DNA? Phosphates and pentose sugar. Next one is base. Okay, that is also called as nucleotide base. Okay, so here you see what is a nucleoside and a nucleotide. Here you see, in case of the DNA, the pentose sugar, that is also called as deoxyribose. In case of the DNA, what is the pentose sugar? Deoxyribose sugar, deoxyribose. But in case of the RNA, instead of deoxyribose, that is a pentose sugar. Pentose means there are 5 carbons. Based on the number of the carbons, we classify the sugars into trioses. If there are 3 carbons, triose sugars. There are 5 carbons, that is the pentose sugar. If there are 6 carbons, that is the hexose sugar. Okay, so the example for the pentose sugar is ribose. So ribose is in 2 forms. Deoxyribose, that means one oxygen is less in the DNA. Ribose sugar is present in RNA. So here, whatever, the question is, what is nucleoside? That is the pentose sugar plus base. Both are called as nucleoside. When these two are attached with the phosphate by phosphodiester bond, that is called as ester bond or diester bond, phosphodiester bond, then it is called as nucleotide. Okay, so nucleoside means without phosphate, nucleotide means with phosphate. These 3 are called as nucleotide. Only these 2, pentose sugar and base, nucleotide base, is called as nucleoside. So that is the answer here. See, a nucleoside consists of here pentose and lipid. So there is no lipid in the DNA or RNA, and hexose and nitrogen base. There is no hexose in the DNA. Pentose and nitrogen base. Yes, it is the right answer. Pentose. Okay, pentose, ribose. It may be ribose or deoxyribose. Commonly, it is called as pentose sugar. Okay, C5H10O5 is called as, that is a formula of ribose. C5H10O5, that is ribose. If it is C5H10O4, that means instead of O5, if it is O4, one oxygen is less, that is called as deoxyribose sugar, that is present in the DNA. So for this question, it is a very simple question. For the answer is pentose and nitrogen, that is called nucleoside. If it is nucleotide, what is the answer? phosphate plus pentose plus nitrogen base. That is called as nucleotide. Okay, so you should distinguish between nucleotide and nucleoside. Next, the question is the membrane bound sac that is filled with watery fluid called cell sap is. So in the cell structure, we find several organelles. Okay, a membrane, it is a single membrane. Usually you see glycosomes, vacuole, and spirosomes. These are all the peroxisome. All these are present in the cell. And in the peroxisome, there is no cell sap. Okay. In the glycosomes also there is no cell sap. And in the vacuole, there is a cell sap. Usually it is called as repository of the cell. Whatever the excretory materials, because there is no excretory system in the plants. Usually in the plants, we find large vacuoles which contains a fluid with the several secondary metabolic products or several excretory substances. It is filled with the excretory substance that is called as that substance is called as cell sap, canarasamon. So that is present in vacuole. That is the right answer. Next, who discovered lamp brush chromosomes? Okay, so we see there are two types of the chromosomes which are called as special chromosomes, which are very large. So they are called as gigantic gained chromosomes. Gigantic chromosomes are gained chromosomes. So there are two chromosomes. One is lamp brush chromosome. Due to the appearance of the chromosome in the form of the lamp brush, these are called as lamp brush chromosomes. Usually these are present in the oocytes of salamanders or reptiles. Okay, so we find this one lamp brush chromosomes which are first described by Fleming, and another special chromosomes which are called as the polytene chromosomes. Polytene chromosomes are present in the salivary glands of Chironomus fly larvae. So they belong to dipterian insects. So in the insects belong to dipterian class. Okay, in the salivary glands, these chromosomes, polytene chromosomes, are present. Okay, so which are first observed by Balbiani. You know, there are some ring-like structures present. These are named as Balbiani rings. Okay, so you should remember these two special chromosomes with respect to competitive exams. One is lamp brush chromosome discovered by Fleming, and polytene chromosomes discovered by Balbiani. And lamp brush chromosomes are present in the oocytes of salamanders, and the polytene chromosomes are present in the salivary glands of the dipterian class of insects. Okay, so you remember. Next, the right answer is Fleming. Fleming, he first observed these lamp brush chromosomes. Next, RNA exhibiting cloverleaf model is. So different kinds of the nucleic acids are there. DNA, RNA. RNA, DNA is again divided into zDNA, like that. Several here aDNA. So here RNA is three types. One is tRNA, mRNA, rRNA. sRNA, actually snRNA, small nuclear RNA. It is a different type of RNA. We need not bother about that. So tRNA was first explained by Halley. It is also called a Halley model. He first described it in the form of a clover leaf. So you see once the structure of the tRNA explained by Halley initially in the form of the clover leaf. Is a cross shape. So this is the clover leaf model of tRNA. You should remember this. There are some specific important points. Okay, here you see 3' end and 5' end. 3' end contains CCA, which is required for the binding of the amino acid. The CCA end of the tRNA, because all these we are not just discussing one question. They may ask several questions from a single topic. See, what is the CCA end? This is the 3' end. It is required for the attachment of the amino acid. The main function of the tRNA is to carry the amino acid for the protein synthesis site towards the ribosome. It is just like a transport vehicle. It carries the amino acid. So it binds to the amino acid with the help of the CCA end, that is 3' end. And there is one more opposite to the CCA end. There is one structure that is called as anticodon. Okay, this is a stem. This is also called as stem structure. Okay, because there are two strands bonded by hydrogen bond. So it is the opposite to the CCA end. There is one more stem like structure, that is stem and loop. The open structure is called as loop. Okay, the structure, the ladder like structure with the hydrogen is called as stem. Okay, so here this is called as anticodon loop. What is the function of the anticodon loop? It reads the codon on the mRNA. tRNA binds to the mRNA to a specific codon with the help of the, because it is called proofreading. It recognizes the codon on the mRNA. The CCA end carries it binds to the amino acid, and when it reaches the ribosome, and between the small ribosome subunit and the large subunit, there is mRNA. On the mRNA, there are codons. So this tRNA binds to the one of the codons with the help of the anti-codon site. Okay, and there is a DHU loop at one side, left side. This is also called DOM or DHO. Okay, so because there is a very little time, 3 minutes, so we have, because I want to explain it everything, so it takes a lot of time. So the main experimental plant of Mendel, Gregor Mendel. So Gregor Mendel, is the father of genetics. What is the plant he used? We know everything. That is Pisum sativum. Wow, how many chromosomes are present in the Pisum sativum? There are 7 pairs of chromosomes. That also you should remember. And jumping genes or transposons were discovered. It is very important. Barbara McClintock. So Barbara McClintock, she was very famous for her discovery of transposons or jumping genes. In the corn, she observed this. Okay, in the maize or corn, she observed this jumping genes are transposons. But because these genes are famous for jumping from one place to another place in the chromosome, so she called them as the jumping genes or transposons. Barbara McClintock. And a cross of F1 with either of the parent is known as, because you see there are 2 parents, okay, parent 1 and parent 2, they are crossed. Okay, and after crossing they produce the first generation, that is called as F1 generation. So that F1 generation produces some gametes. Okay, when it is crossed again with the one of the parents. So there are 2 parents. One is the recessive parent, another is the dominant parent. Homozygous dominant and homozygous recessive parent. Okay. So what is that called? Either of the parent is called as back cross. Okay. But if it is specific to the recessive parent, if the F1 is crossed with the recessive parent, that is called as test cross. If it is crossed any of the parent, you should that's why you should study the question very well. If it is crossed with F1 is crossed with any of the parent, that is called as back cross. So that you should remember. And the initiation codon in protein synthesis, you should know, that is AUZ. That is called as initiation aramba codon. Usually most of the times it is AUG, but in some of the bacteria that is a GUG. Okay, so AUG or GU is the right answer. So when the codon of amino acid is mutated into a termination codon, it is known as. So in the mutations, there are different kinds of the mutations that I discussed here. That is the, here you see, that is the sequence here. Silent mutation. What is the silent mutation? There is one nucleotide, there is a change in the base, okay, nitrogen base, but that doesn't change the amino acid after protein synthesis. Even though one of the nitrogen base is changed, that means there is a mutation, but that is not reflected in the protein. Okay, there will not be any change in the protein structure because the same amino acid is produced even though there is a change. See, instead of GUA, here there is GUU because both the codons code for the same amino acid, that is valine. So that is called silent mutation. And what is missense mutation? Missense mutation means here